All right, so we're gonna try to cover three things today. So today in particular, I probably won't ask the audience any questions because that always takes a long time for you guys to answer. We don't have a lot of time. We're gonna try to hit three subjects today, but because I'm not asking any questions, if you have any questions, still feel free to just like raise your hand and say hey, and then I'll answer your question. So we're gonna have a review session for exam 4. This same time on Thursday, so it's your normal class period, and it's a reading day, so everybody should be able to make it. So at the review session, I'm just standing here, and I got all my PowerPoints ready, and I'm just answering questions. And when the questions stop, the review session's over. So if you have some time to review your notes between now and Thursday, you can just bring it in and bring in any oh, I don't understand this thing, what do you mean by that, or whatever, any questions about it. It's not going to be a lecture. It's just going to be answering questions, and and don't feel shy because generally people just show up. They don't have any questions, so there's virtually nobody here, and then we'll leave in 30, 40 minutes. So if you have questions, just keep asking them, and I'll answer any questions that you have on Thursday. It'll be like a review session just for you if you ask questions. If you don't ask questions, you'll be at the mercy of people who ask questions. Okay, so that'll be this Thursday. All right, so we're going to talk about apoptosis now, and there's two types of cell death, necrosis and apoptosis. So necrosis occurs generally when cells are damaged. This causes the cells to swell and then burst, so all of their contents get released, and if you're like a multicellular animal, what will happen is those cytosolic components, the parts inside the cell are now outside. They're going to cause an inflammatory response as your immune system says, hey, there's some cellular damage in this vicinity. Okay, but we're not going to be talking about necrosis, just as a contrast. We're going to instead be talking about apoptosis, which is also called programmed cell death. And apoptosis comes from the Greek word meaning dropping off or falling off, as if falling off, like leaves falling off a tree. So every fall, the deciduous trees lose their leaves. Trees not dying, the leaves are, and but it's all to help the tree survive. And the same way, apoptosis is a natural process that that a multicellular organism wants to kill some cells, and it's going to go through this pathway. Okay, so it's going to occur in response to normal extracellular developmental signals. And it's going to occur by a defined molecular pathway. There's a few little branches in there, but it's generally one pathway. And then nothing's really wasted because neighboring cells are going to engulf the dying cell and then reuse all the components. And because the cells that are dying are being engulfed, there's nothing released into the cytoplasm, and so there's no inflammatory response. Okay, so these are just a few of the ways that programmed cell death or apoptosis benefits the organism. So one is the maintenance of adult tissues. So you basically you're making cells, you have to turn them over, at a certain rate. And the most obvious here are the blood cells, where an average human is making 500 billion, 5 times 10 to the 11 blood cells every day. And so if you're making that many, well, you have to get rid of that many too. So you're going to be killing that many by apoptosis. Then there's the elimination of virus-infected cells. So one of the ways that your body limits viruses infecting is just to kill those cells. So the cells, when they're virus-infected, will kill themselves. And while that's pretty drastic for the cell, it helps the virus from spreading, or it helps the body by limiting the spread of the virus. Okay, and then similarly, if a cell is very damaged by DNA, then the cell is going to just cut off and decide to kill itself rather than try to repair everything and not all the DNA damage and not fix everything and then have a chance of, let's say, activating oncogenes or inactivating tumor suppressor genes and then causing cancer. Okay, so it's a way of it's better for the whole body to lose a few cells that were very damaged rather than have that cause cancer. Okay, and then during the immune, you're creating your immune system. So when B cells are making antibodies or T cells are making T receptors, they're basically modifying the sequence, and then they're making them in they have lots of little components which they're rearranging to make the antibodies and the T cell receptor. And so this is going to produce new combinations, and then they're going to test whether those new combinations recognize self. And those so when you get a new T and B cell, they're called immature, and they're going to mature for B cells in the spleen or for T cell receptor in the thymus predominantly. And at that point, they're being exposed to peptides that are coming out of the body, which are, this is more information you need, but it's being co-expressed with an MHC molecule. So they're looking at that combination, and then if they see, something like if their antibody or their T cell receptor responds to that, then they're going to kill themselves. Okay, because if they didn't do that, then they would go out in the body and they start attacking the normal cells. So about 95% of the immature T and B cells that are made are going to die based on self-reactivity, okay? But if you didn't have this, of course, your own immune system would be attacking you all the time. Then there's the sculpting of tissues during development. I'll show you a slide on this. Basically, you're born with webbing in between your digits. All that's going to go away. And that's just one example. There's like tadpoles, I'll show you that too. And then nerves. So there's, a lot more nerves are made than are going to survive. So basically, nerves often have to travel long distances, sometimes from the spinal cord out to the end of your arm or your legs, or they're going from your eye into your brain, and they have to make the right connections. So if they don't make the right connections, then they're going to be killed. Okay. So one thing that's occurring even probably in a lot of you right now, the tail end of it is during puberty, your prefrontal cortex grows like crazy. And then during the ensuing years, till about 21 or so, it just keeps rearranging itself, and you get apoptosis as it's making the right connections. So this is like, a constant thing. There might be tail end of that, but it basically allows you to have, some sense of restraint. Like, should I really be yelling, like crazy at this party? Like, yahoo, like you do, like your early high school, and you're like, what are they doing, Well, because they haven't developed their prefrontal cortex yet, I would say. Maybe you could just say they're immature, but it's about the same thing. All right. So anyway, here's some of these examples of the developmental things. So like you can have like these webbing in between the mouse paws, and you can see that, this can be manipulated, right? So like you can have a duck, and the duck doesn't kill that, right? So the duck has webbed hands, so it can paddle well, but like a crow, it would lose it, right? So it has these, defined feet. So you can, these kind of things can be manipulated. The tadpole tail, the whole tail is apoptosed to give rise to the frog. So, but you could imagine changing that now that the frog would have a tail. So it's like, these are developmental uses of apoptosis, and they can vary between species, of course. All right, so these are some of the events that would happen if you looked at an apoptotic cell. So what you would notice is chromatin starts to condense, and DNA starts to fragment. And then it gets cleaved, right? The fragmentation is just cleavage. There's a DNase that's cutting it up. And as you know, there's you have nucleosomes with about 200 base pairs wrapped around the nucleosomes going to the next one, and then it's wrapped around, and you get 200 going to the next one. So when this thing cuts, it cuts at a site where it's connecting, and so you end up with like if you cut 4 of them, it's 4 times 200, or 3 is 3 times 200. So you end up with, if you were to take this and run it out on a gel, you'd get a DNA ladder, which is every 200 base pairs, and that's a sign of apoptosis, because it's DNA is being cut up in multiples of nucleosomes. Okay, then the nuclei, so you're going to get this chromosome condensation, and it's going to go to the periphery of the nucleus. Then the nucleus is going to fragment, and then by this time, that cell is generally engulfed, okay? It's just being taken up by the neighboring cell. But let's say you were doing this in tissue culture and there's no cells nearby to engulf it, or you induce them all to kill themselves, then what will happen is, even the cell will fragment. We call this blebbing, where it like puts out parts of itself. But this blebbing generally does not occur in the animal because it's already engulfed before that step. So here we're looking at a normal cell nucleus. Here's a normal cell. Here's the nucleus. You can see some condensed. So this is electron dense area. This is a transmission electron micrograph. So this is just chromatin. You get a little bit, some condensed chromatin on the ends, like heterochromatin, or, I mean, who knows? This could be the bar body in a female, where the inactivated X chromosome. But generally, the chromatin is spread out here. A lot of it you can't see. But then you look at the apoptotic cell. This is the nucleus, this is the nucleus here. It's blebbing. The nucleus itself is blebbing. And if you look in there, you see this super dark area. Well, that's the condensed DNA. So the DNA is condensed, going to the outside of the nucleus, and the nucleus is starting to bleb out. Okay, so the nucleus, remember, the nucleus fragments first, and then the cell blebs out and fragments later. Okay, but as I said, before the cell can fragment out, it's going to be engulfed by a neighbor. And the signal for this is, so almost all the cells in your body have a receptor, and that receptor is looking for cells that are killing themselves. And so what will happen is the cell that's going to kill itself puts up on its surface a receptor that says, hey, I'm killing myself by apoptosis. Those two, so the other cell, which is looking for that, will see it. They'll have cell-cell communication through this receptor, and then that cell will get targeted. So the apoptotic cell is actually putting a molecular signal on its surface that says, hey, I've decided to kill myself. And generally, almost all your cells in your body have receptors for that signal so that they can engulf cells that are near them that are killing themselves. And then that gets autophagous, turned into like a big phagosome, and then just gets recycled. So you didn't actually waste that. I mean, you did waste it in a way because you have to, repurpose all those things. But it didn't just get like spilt out into the cytoplasm or like, the space in between cells. Not cytoplasm, space between cells. All right, so here again are some pictures of a necrotic cell. You can see the cell swelled and burst. All of its contents are gone. And then apoptotic cell, again, you've got this blebbing nuclei with a condensed chromatin. And then here's an engulfed apoptotic cell. And you can see you got engulfed before it started blebbing, and you got the condensed chromatin on the nucleus. But this whole cell is inside of a larger cell. All right, so this whole pathway was worked out in C. elegans, and while they were giving out the Nobel Prize for creating C. elegans as a model system, they also threw one out to Bob Horowitz, who had done a lot of the work in this pathway. Though my advisor actually identified the first cell death mutant, which basically was in that, it basically perdured because it didn't put out the engulf me signal. That signal, that they're put out when they're, it was mutated in that. So you would just have apoptotic cells hanging out. And so here you can see, C. elegans. So C. elegans was easy to study this because it's transparent. And cells that are killing themselves, when you're using Nomarski microscopy or DIC microscopy, you can look at a particular layer. And these cells look like they're sticking out of the plane. And so they look like little buttons sticking out. And so like my advisor identified a mutant where there'd be a ton of these things hanging out because they couldn't be engulfed anymore. And then a SED3 mutant, as we'll see later, this is one of these caspases that's going to be doing the apoptosis. And if you don't have this, these cells, the same cells don't kill themselves. And this is one of the reasons it's called programmed cell death, is because in C. elegans, it's an invariant lineage. All the cells divide at the right time. They become the right thing. And in certain parts of the division, this cell is destined to kill itself every time. And so it's programmed. The cell death is programmed. That cell is going to be made. It's going to immediately kill itself. So you know when the cell is supposed to kill itself and, at what time. And so it's very easy to study whether or not this is happening or not, because it's always supposed to happen at exactly the same time in development. All right, so here's this overview of this pathway. It looks a little complicated for now. So what we're gonna come back to this, and we're gonna what we're gonna do right now is we're just gonna focus on the fact that this pathway is gonna eventually lead to upstream caspases, which are proteases, activating downstream caspases, which are related proteases, and those proteases are going to chop up proteins. Okay, so activating these caspases, these proteases, are going to cause downstream proteins to get chopped up, and that chopping up those proteins is what causes apoptosis. All right, so we're going to be interested in how the upstream caspase gets activated and how it activates the downstream caspase. So all these things up here are going to be focused on activating the upstream caspase. Okay, so what happens when the caspase gets activated? So you can imagine different scenarios. So not all of them are to inactivate the protein. So here, activity is shown by this yellow halo. And you could have a monomeric protein, a single protein, and it gets cut, and now it was active, now it's not active. You could have a multi-protein complex with multiple proteins, and you could cut them, and they were active, and now they're not active. But you can also have other scenarios. You can have a protein that's inactive, and then when it gets cut, it now becomes active. And then you can have another protein that's an inhibitor of a protein. So the target protein's inactive because it's got an inhibitor. So now you hit the inhibitor, you inactivate the inhibitor, and it releases the active protein. Okay, so all these things. So certain proteins are going to be activated by these cleavage events, and certain proteins are going to be inactivated, and they're all part of a pathway for the cell to kill itself. So basically, a whole lot of downstream proteins are going to be modified by these caspases cutting. And remember we had talked about the ubiquitin pathway, right? And it gets a substrate and it just turns into little peptides. That's not what we're talking about here. We're talking about proteases that have a defined sequence they're looking for, and they just cut it right there. Okay, so they're only going to cut certain proteins at certain positions, and that's either going to inactivate the proteins or it's going to lead to their activity. So one example of this one here, where you have an inhibitor with a protein that's inactive, and then during apoptosis, the inhibitor gets cut, is the ICAD-CAD system. So ICAD is the inhibitor CAD, okay? So ICAD, it would be like this protein. CAD is the protein that's being inhibited. Well, what is CAD? CAD is the DNase that's chopping up the DNA, okay? So basically, in your cells in your body, there's a protein that, if it got released, would chop up all your DNA. And it's just being inhibited all the time, just waiting for when apoptosis occurs. When apoptosis occurs, the protein that's inhibiting it gets cut. It releases the DNase, which now chops up all the DNA in the cell, okay, into those fragments. So it's sort of scary, right? It's like you got a bomb in every cell in your body, waiting to you get activated, chop up your DNA. But that's how it actually works, right? Your cells are constantly poised to kill themselves if they get the right signal. All right, so now we're going to be talking about the upstream caspase activation, or what's happening. So, well, first off, the way the upstream caspases activate the downstream caspases is they're going to cut it. All right, so there's a pro-domain that's going to get cut and removed. Then it's going to cut the downstream caspase in two. But both pieces are going to assemble together. And in fact, it'll assemble as a dimer, and now you have the activation of the downstream caspase. Okay? So this is how all the downstream caspases get activated, by cutting them. Okay? So now there's two ways that we're going to talk about to activate the upstream caspases. That's the caspases that are cutting these downstream caspases. All right? So there's induced proximity. So what happens here is there's 3 binding sites, and it can bind 3 of these caspases that then will cut each other. These are upstream caspases. And then they assemble into, with, again, looks like a dimer with the 2 pieces self-assembling, looks like the downstream caspases. So what happens here is you've got 3 of these binding sites brought together. And we'll talk about this one at the end of the lecture. Okay? Then you've got another way. So this is all to make the upstream caspase. This one's called caspase 8. And then there's another one. It's for caspase 9. And these are the only 2 upstream caspases we're going to talk about. Caspase 9, and they're the main ones. Caspase 9 is going to get activated by the formation of a huge complex. Okay, so it's called a holoenzyme. Holoenzyme just means there's a lot of subunits. And it's made from APAF1 subunits. And those APAF1 subunits can only assemble if they're bound to cytochrome C, which is normally involved in the electron transport chain. Okay, so it's normally moving electrons in the mitochondria. Okay, that's its day job. But it will get released into the cytoplasm, and when it gets released in the cytoplasm, it won't do its day job. Now it's going to bind to APAF1, and that's going to make the APAF1 assemble into a holoenzyme called the apoptosome. Okay, so it's like a big complex called the apoptosome, and some of these drawings are incorrect. So like this one looks like there's 6 units, but it's not. Okay, there's 7. It's a 7-mer. And it looks like the caspase is bound on the end, but it's not. The caspase is bound on the inside. I'll show you in a minute. So this is like the APAF1 by itself. So it's self-binding to itself, and there's no way it can form anything because it's bound to itself. I don't know what that is. Do you? It's what? Oh. All right, let's go back here. That's annoying.
My text you will show up too.
Don't do it. Nobody texts me. Hopefully you don't know my phone number. But I did turn off the noise. Anyway. Okay, so this thing is folded on itself, so it's not doing anything. But if cytochrome C binds to it, that causes it to not fold to itself. Okay, so cytochrome C is releasing the self-binding of the APAF1 subunit. So cytochrome C opens it up. Now that the APAF1 is opened up because it's bound to cytochrome C, it can assemble into a 7-mer. Yeah, just like shown here. And that thing is that is the apoptosome. Okay, and that's going to bind 2 of the upstream caspases, which are caspase 9. Okay, and this is a structure that came from studying C. elegans. So in C. elegans, the apoptosome is made with 8 subunits, and it looks like this. So we're looking at it from the top. Now we're looking at it from the side, and now we're looking at it from the bottom. Okay, so what you see is that from the top, you can't see the hole. From the side, you can't see the hole either. But at the bottom, you can see that it has a little cavity. Like if you just look at it if you look at this one from this direction, it's got a cavity, and in the cavity is where the 2 caspase 9s sit. Being in that cavity activates caspase 9. Okay, so remember, this whole thing could only assemble because cytochrome C was released, and then when cytochrome C is released, now it assembles, and now it can bind 2 caspase 9 to activate them. The mammalian one, which is just modeled, this one's actual crystal structure, and this one's a modeling, but it's got 7 instead of 8, not a big difference, right? This is look from the side. And then it also, here we got a cutaway view. It's not from the bottom. Now we cut it away from the side. I don't know why they're doing differently. And you can see that from the side view, it's got 2 caspase 9s in it. Okay, but again, it's activated the same way. And so this is all in response to the thing assembling when cytochrome C is around. Okay, so we've been working backwards, okay? We've been working backwards from the upstream caspase, Cas9, to the apoptosome, forming from APAF1. Okay, APAF1, and that was only able to form because it was self-binding. It could only form when cytochrome C was released. So cytochrome C bound it. Now it could form that a big apoptosome, and 2 caspase 9s could bind and get activated. Okay, So now what we have to focus on is, well, how does the cytochrome C get released? Because not normally cytochrome C is in this inner mitochondrial space right here, okay? Well, how's it going to get out, okay? So now we're going to focus on how cytochrome C is released to activate the apoptosome, APAF1, to form the apoptosome, to activate caspase 9, okay? And we're going to be looking at these components, BCL2, which is anti-apoptotic, and it's got related proteins called BAX or BAK, BAX, BAK. These things are pro-apoptotic, and they're going to be involved in releasing cytochrome C. All right, so BCL2 was identified actually initially from cancer. There were some mutations that increased its expression. And that caused white blood cells that were normally, like you normally have to kill billions of them a day. But these white blood cells had BCL2 overactive, like overexpressed, and they were not dying. And that right there was causing leukemia, okay? So to get leukemia in blood cells, all you have to do is just prevent them from killing themselves. Because they're still going to be made, billions of them are going to be made every day, but you're not killing them, so now you're going to end up with billions too many cells. Every day you go by, you get billions of extra cells. Okay, so that's how it was identified, and we're going to get into how it works, but first we're going to look at its structure. Okay, so there's three classes of these proteins. There's these pro-survival things, and that includes BCL2, we just talked about, BCL-XL. So these are the two major ones that you might see in these slides, but there's more. Then there's the ones that cause apoptosis, pro-apoptotic. And then the two we're going to talk about are BAX and BAK. But these are almost interchangeable, and these are almost interchangeable. We don't need to worry about them too much. And then there's the BH3 only, and here we're going to talk about like BIM and BID and BAD. Or maybe we'll just talk about BAD and BID. We'll see. We'll see what's in the slides. All right, so you can see they're all sort of related to each other. So they all have a BH3 domain. But the BH3 only pretty much only have a BH3 domain. The pro-apoptotic have a BH1, BH2, and BH3 domain, and the pro-survival have those same three plus a BH4. So they're all coming from the same ancestral gene, and these are duplicating gene families. But as they've duplicated, they've picked up different roles. So these are going to prevent apoptosis. The middle ones are going to cause apoptosis. And the BH3 only are going to initiate apoptosis, okay? And we'll see what they're doing in a minute. And they all have this transmembrane domain, so they can all go into membrane. Okay, so here we're looking at some BH3 only proteins activating this pathway to release cytochrome C, to bind APAF1, to form the apoptosome, to activate the upstream caspase that then activates the downstream caspases, okay? So we're still interested in how the cytochrome C is released, and this slide does not tell us that, okay? It's just like a picture of the process from a big view. So now we're going to get into how is the cytochrome C released based on these proteins. So this slide is critical, okay? So this is the critical slide that explains how it goes. Some other slides are going to have misinformation. This is the real slide. So if you have any confusion about what the correct slide is, it's this one. All right, so what's going to happen is the BH3 only protein have to get activated through some mechanism. Maybe they were inhibited, and the inhibitor has to be inactivated. Maybe they're inactive to begin with, and you have to get cut. Something's going to happen to the upstream protein, the BH3 only protein, to activate it. Okay, so then what happens? So ultimately, we're interested in how cytochrome C gets released from inside the mitochondria, which is this bottom part, to outside the mitochondria, which is up here. And the way they're going to get released is the pro-apoptotic proteins, when they're in the plasma membrane, are going to multimerize to form a huge channel. So they're going to bind each other, and as they bind, they exclude the plasma membrane on the inside. So we end up with a big hole. in the mitochondria. So like this is what it looks like. So here we're looking at fragments of mitochondria, and here in green is Bax. So this is one of those proapoptotic things, and you can see that like, and here this time 22 is embedded in the plasma membrane, and you can see there's no plasma membrane where this thing is making the huge hole. Okay, just a humongous hole in the outer membrane, and cytochrome C and anything else in there can just come straight out because it's a big hole. Okay, so let's go back to our slide. Now, what we're interested in is how this thing can form. Well, okay, so these proteins are generally not even in the membrane to begin with. These proapoptotic like Bax or BAC. What will happen is the BH3 only is required to put it into the membrane. Okay, so this is an upstream thing that's going to be required to put in the membrane, and then they can multimerize. But then what are the anti-apoptotic proteins doing? Like BCL2 or BCLXL. Okay, what they do is they're actually not they're sort of in the membrane, but they're not fully in the membrane. Okay, like they have one transmembrane domain, but the BH3 only protein will put the other membranes, like here you see membrane 9 in the transmembrane section 9, but then 6 and 5 are out. So BH3 only is going to put it in the membrane. Okay, so BH3 only is going to activate the proapoptotic, but the BH3 protein is also going to activate the anti-apoptotic. So what is the anti-apoptotic doing? It's trying desperately to prevent the proapoptotic from forming this big multimer. And the way it's going to do it is it's going to bind them one-to-one. Okay, so if it turns out that you have more anti-apoptotic than proapoptotic, all of the proapoptotic will get bound up and can't form the big channel. Okay, so that's how it works. So while you could think that the BH3 only is proapoptotic, and it sort of is because you can't get apoptosis without it, right? It initiates it. It's activating the proapoptotic by putting it in the membrane, and it's activating the anti-apoptotic by fully putting all of the transmembrane domains into the membrane. Okay, so it activates both branches, and then whoever has more will win. Okay, if there's more anti-apoptotic, no apoptosis occurs because they can't form this channel. If there's more proapoptotic, then yeah, some of them will get bound up by the anti-apoptotic, but there will be enough left over that they'll form that channel. Okay, so that's what determines it. All right, so we're going to look at particular examples of how cell death is regulated. One example are these neurons. So like I told you, the neurons have to hit their target, and your body, because they're often going over big distances, your body's going to make more neurons than it needs because it wants to make sure it hits a target. And who knows, that neuron may take a bad turn or something, right? So the nerve cells, you make more of them, they're going to hit their target. The target cells are going to give them a survival factor that will keep them alive. And that's called a neurotrophic factor. Okay, so those are factors that keep them alive. And they get it from the target cell. So if a nerve cell put out its process over here, well, there's no neurotrophic factor, it's going to kill itself. Like here it looks like they all hit it, but in fact, once one of them makes a connection, the other one really can't get at that particular target cell. But at any rate, the one that does not get the connection, they end up killing themselves. Okay? So your body's making more nerves than it needs to make sure it hits every target that it needs to hit, and then it kills the ones that did not reach the target or bind to it sufficiently. Okay? So we're going to go into what is the pathway, the molecular pathway by which a neurotrophic factor controls the apoptosis. All right, so here's a diagram. Now down here, just ignore all this nonsense about ions going in. This is what happens when you take old reviews. That's what people thought was happening. That was ridiculous, right? I mean, it was like speculation. No, remember, go to the other slide if you want the mechanism. The mechanism is Bax forms huge channels that everything just comes out. It has nothing to do with ions going in. It's nonsense. I just like this slide because it's got a good drawing up here. So we're focusing on the upper part. Okay, so what's happening? So when the neurotrophic factor is present, the PI3 kinase gets activated from this receptor. It's going to activate AKT kinase, and then AKT is going to phosphorylate a particular BH3 only protein called BAD. That phosphorylation is the signal for a 14-3-3 protein to bind to the phosphorylation and inhibit BAD. Okay? So what's happening? The way these nerves keep themselves alive is they're constantly getting the neurotrophic factor, which is causing this pathway through PI3 kinase to AKT to phosphorylate BAD to keep it inactivated. Okay? So it's being inactivated. Now what happens if there's no neurotrophic factor? Well, PI3 kinase does not get activated. AKT does not get activated. BAD does not become inactivated by this phosphorylation. So BAD then comes in, and again, just ignore this part of the talk, the slide. BAD comes in, and it's going to take the activating pro-apoptotic and put it in the membrane. It's going to take the antibiotic and put it in the membrane, fully put it in the membrane, and then they're going to fight it out to see whether you get apoptosis or not. Okay, so that's how it works by going to BAD. But even this drawing, which I like, is incorrect. If you guys remember from, did they go over PI3 kinase? Huh? Did you guys go over PI3 kinase? You did? Okay. So if you remember, PI3 kinase is not working like this in the cytoplasm. It's all happening at the membrane. This just to remind you that you got to take these pictures with a grain of salt. So basically, as you know, this is a receptor tyrosine kinase. It's going to cross-phosphorylate based on the neurotrophic factor. You're going to PI3 kinase is going to bind, which brings it to the membrane. PI3 kinase job is to take PIP2, phosphorylate it to turn into PIP3. That's the docking site for AKT and PDK1. They're both kinases. PDK1 then phosphorylates AKT. Now that AKT is phosphorylated, it can go away from the plasma membrane and becomes activated. So this is all clarification. The clarification is that this slide was sort of, playing loose with the pathway by showing you it in the cytoplasm, when in fact it's all happening at the membrane. Okay. All right. So we're going to go to another type of cell death now that's going to use a different upstream caspase. This is caspase 8. And this one is the induced proximity pathway. All right, so what are we looking at? We're looking at cytotoxic T cells. And let's say this cell were infected with a virus. It's going to chop up the protein, put it on the surface, and this cytotoxic T cells, hey, I recognize a foreign protein with your MHC protein, which we don't need to go over in this class. And it says, hey, you've got foreign proteins inside of you. Probably there's something wrong, like maybe you've been infected. So it's like, I'm not taking any chances. I'm going to kill you. So the cytotoxic T cell is going to send a signal to kill this cell. All right? So what is that signal? Because it's not killing it. It's just telling that cell to kill itself. It says, hey, I'm going to give you the signal to kill yourself. So what is that signal? Turns out the signal is just putting this thing called fast ligand in 3 of them all together. Okay? So what's going to happen, let's draw this out. So the cytotoxic T cell, when it's made a determination, it's going to kill the target cell. It's going to put out on its surface 3 of these binding sites or ligands next to each other, and it's going to make them all go in the interface next to where this target cell is. It doesn't want to kill just every cell around it. It just only wants to kill this cell. And it's going to put these in this domain, in this area. Okay? These will get bound. So these things were originally just floating around. But because there's 3 of them now, they'll bind to the 3, okay? And the fact that these things which were just floating around are now localized right next to each other, 3 of them, that's the signal. So it's sort of like receptor tyrosine kinase. The bringing them together is the signal. And in this case, bringing 3 of them together is the signal. And this is, of course, this isn't secreted. You can get some of these secreted, but here the cytotoxic T cell wants to kill this particular cell, so it's going to put it on its plasma membrane, right? It's putting it on its plasma, so it doesn't diffuse around. It doesn't want to kill every cell in that area. It's going to do this. So there are some situations where you will get these soluble ligands that are triples to kill things, but that's not what we're dealing with now. All right. So what happens? So you got the fast ligand, 3 of them. The ligand just means it can be bound to the receptor. And for cytotoxic T cell, these are connected to the plasma membrane of the cytotoxic T cell. So then fast is the receptor that binds the fast ligand, and you get 3 of them. So remember, before they were not together. They're now binding them, and they come together. So you get 3 of them together. These fast receptors are associated with an adapter protein that can bind caspase 8. Caspase 8 is the upstream caspase. So it can bind 3 of them, and what they're going to do is they're going to cut each other, the caspase 8, and then again you get that formation where you've got it's cut and then it reassembles 2 cut ones together, and that's the active caspase 8 that's going to cut the downstream caspases. Okay, so the way the cytotoxic T cell has killed the cell is by putting this triplet fast ligand on the surface next to it to allow the fast receptor, which is already expressed on the cell, on every cell. They're just waiting to get the signal to kill themselves. And then said, oh, I guess, the cytotoxic T cell is telling me to kill myself, so I better kill myself. And that's what's happening through this pathway. So this is a sort of complicated slide, but it illustrates an important point that so this one's called, the one we just went over, and this is, again, you can just call this fast ligand, and then this fast receptor. It might have different names here, but there are other classes of fast-like proteins, okay? But for this class, we'll just call them fast and fast ligand. And then this adapter is called Fab. So it's basically the same as this whole thing. It's just got different names. Okay, so it's going to bind the procaspase 8. They're going to cut each other, and then you get that active thing where the part that gets cut will self-assemble the 2 pieces, and that's the active one, that will then take the downstream caspases and cut them, and then they'll become active, right? That's the pathway, okay? And then these ones will then start chopping up cellular proteins, and that's how you get apoptosis, okay? But it turns out that that there's these things called inhibitors of apoptosis. So there's actually a number of places where, the cell doesn't want this to just happen by chance. So it's got things in there that are like buffers that are just preventing this from happening unless you get a ton of signal. So one of these things is this flip. What is a flip? A flip looks like caspase 8. It can bind in there too. So if you were to get a flip in there, they can't cut each other. Okay, so it's like a buffer, right? You got to get a whole lot of these things so that you use up all the flips. Okay, because why? Because the cell doesn't want to just kill itself for nothing, right? Like you just happen to get these 3 together, and now the cell kills itself. That's not good. So you got a buffering system. Okay, the flip is you got to dilute it out because it's a competitive inhibitor for this pathway. It's going to look like caspase 8 and block it. Okay. So then there's another inhibitor. There's another buffer that's going to prevent this from happening. There's these inhibitors of apoptosis, or IAPs. They're going to prevent the procaspase from getting cut to the active caspase. These are the downstream caspases, okay? They're going to prevent the upstream caspase from doing that, okay? So that's another buffering system, because you don't want to just have some rogue upstream caspase activate these guys, and then suddenly you've got, the whole cell killing itself. So it's being prevented by these inhibitors of apoptosis. And these were actually identified here at UGA by Lois Miller. So basically what she was doing is she had a virus, a baculovirus. It's an insect virus. It infects insect cells. And she was just randomly knocking out every gene on this huge virus and say, well, what does it do? And when she knocked out this one gene, she put it into the cell killed themselves. She said, whoa, if I knock out that virus gene, the cell kills itself. Okay, it was killing itself through apoptosis. Okay, so basically what that one gene was doing was preventing the cell from killing itself. Okay, so it was an inhibitor of apoptosis. And then they went on to show that while that was a virus-specific protein, they went on to show that there were cellular inhibitors of apoptosis that were doing the same thing. And other viruses actually use this type of mechanism, because it's like a race, right? Like if a virus infects a cell and the cell kills itself, well, the virus is not going to have any luck expanding itself, right? That virus is going to be out of luck. But if the virus can go in and block apoptosis, now it can expand itself. Okay, so the different ways to do it. So some of them express the actual counterparts of the IAPs. So the first one she identified was virus specific, but then she saw that other viruses actually picked up parts of the gene from that organism that allowed it to block it. So they actually picked up the cellular IAPs and incorporated it into the virus, again, to block it. Other ones overexpress BCL2, which you remember BCL2, right? It's the this one. So it just overexpresses it like crazy. So then the pro-apoptotic can't even form the channel, okay? So the viruses have different strategies, but their goal is to block apoptosis, because otherwise, they're going to die with the apoptotic cell. All right. So we've got this problem where, okay, even if you have enough of the signal, because the cytotoxic T cell has loaded up that surface with a ton of these fast ligands, okay? It outcompeted all of these competitive inhibitors, these FLIP, and now you've got the activation of the upstream caspases. You still got a problem. That problem is it's hard for the upstream caspase to activate the caspases because of these inhibitors of apoptosis, okay? So it's got to get rid of these guys, these inhibitors of apoptosis, to be able to do its job. Okay, so it's going to activate the intrinsic apoptotic pathways. This is extrinsic for external, and then it's going to do the intrinsic, which goes to the apoptosome. I agree this picture of the apoptosome is pathetic, right? It's a big, the big complex, forms of 7-mer, just ignore what it looks like. This is the normal apoptosome, okay? It's gonna get activated again by cytochrome C. So why does it want to open up the membrane to release cytochrome C here? Why, just have more activity? No, that's not the case. It turns out that inside the mitochondria is something called SMAC Diablo, and one lab called it SMAC, and one lab called it Diablo, and they published in the same issue of Cell, and so we have to call it both, because nobody has priority. So SMAC or Diablo, same protein, it's being released. When you make these big channels, it's coming out of the mitochondria. And when it comes out of the mitochondria, it's going to inhibit, its job is to inhibit the inhibitors of apoptosis. So these inhibitors of apoptosis are preventing the procaspase from getting cleaved to the caspase by the upstream caspase. But now, SMAC Diablo, when it's released, inhibits it. And then, of course, at the same time, you're releasing cytochrome C, which activates the apoptosome, so now you have even more activity to do it, okay? So this extrinsic pathway is going to also lead to this thing. And even when you do the intrinsic pathway and you cause a big hole, you're also inactivating the IAPs, because you get SMAC Diablo released, which is an inhibitor of the inhibitors of apoptosis, okay? So then the question is, well, how does this upstream extrinsic pathway activate this, form those big channels in the mitochondria? And the way they do it is they're going to take one of these BH3 only proteins called BID, which is inactive, doesn't work, and they're going to cut it, okay, because they can cut proteins. Now the truncated BID is now active. So what's a BID? Well, we already saw it in that diagram, right? It's a BH3 only protein. These are one of these proteins that are going to take the pro-apoptotic, put them in the mitochondrial membrane. They're going to fully put in the antipoptotic in the membrane, and then you have that fight to see if you make the channel or not. Okay, so what this is saying is that through this activating bid, the extrinsic pathway, which uses caspase 8 through this fast ligand pathway, is also going to open up channels in the mitochondria that, one, release SMAC diabolate to inhibit the inhibitors of apoptosis, and two, bring in the apoptosome, assemble it, which will also end up cutting the downstream caspases. And then the only thing we want to talk about is that P53. Remember how when you have a lot of DNA damage, well, if you have any DNA damage, P53 gets activated. And if you have a lot of DNA damage, P53 is going to signal the cell to kill itself. So how does P53 kill the cell? It does it because one of its transcriptional targets, okay, it's going to assemble into this tetramer, as we talked about before. One of its transcriptional targets is BAX. And then you're like, by now I hope you know who BAX is, but if you forget, you can always go back to the same slide. BAX is one of these pro-apoptotic. And remember, it's always a fight between the antipoptotic and pro-apoptotic. So when P53 makes a whole mess of BAX, you're much more likely to overcome this inhibition and form these big channels. Okay, so that's how P53 promotes apoptosis when there's a lot of DNA damage. Okay, is there any questions about that? All right, very good. We're moving. Moving on. We're going to now do microRNA. All right, so we're going to talk about microRNAs and RNAi. So this was awarded the Nobel Prize a few years ago to two people I know who are in the C. elegans field because it was discovered in C. elegans. Again, another Nobel Prize for C. elegans. It's been like 3 so far. So basically, what this is at its core is if you have double-stranded RNA, okay, double-stranded RNA is going to cause the degradation this is for RNAi, okay, RNA-mediated interference. And the guys who won the Nobel Prize was Andy Fire and Craig Mello. The double-stranded RNA is going to cause that mRNA to get chopped up. And Andy Fire basically got the Nobel Prize for realizing that it was double-stranded RNA that was doing it. And he called up Craig Mello, who had shown that this thing, once you put it in one part of the worm, it gets amplified and spread to the whole worm. And so Craig Mello gets the Nobel Prize because Andy Fire called him up and and told him this insight. But you're basically getting it for the mechanism. But Craig Mello then later did a lot of work to flesh out the pathway. So basically, like here, they're doing an in situ hybridization for a particular gene, mRNA. And then once you do this double-stranded RNA, you see it's gone. Okay, just all got degraded. So this is a way, scientists love this, okay, because this is a way they can inactivate any gene they want by just making a double-stranded RNA against that. That's why they won the Nobel Prize. Not because people care about C. elegans, RNAi, or whatever, right? It's because this mechanism now works in all these different organisms, right? Single-cell organisms, plants. In fact, the plant people were already on this, but they just didn't understand that it was double-stranded RNA. So they just barely missed out on getting the Nobel Prize instead of the C. elegans people. Okay, so it turns out that microRNAs, so RNAi is really a defense system that seems to really be geared against blocking transposons, just chopping up transposons, because you're trying to limit transposons jumping around. So so there's a lot of retrotransposons that go through RNA, and you're going to chop it up. Okay, so this is why this system evolved, and we can use it to chop up genes of our interest. And the siRNA are generally going to bind as a 21-mer or around, and it's going to bind completely, and it's going to cause the target to get chopped up. Now, the cell also has thousands, hundreds or thousands of microRNAs. Now, these are like genes that produce a microRNA. And the microRNA is going to target a particular mRNA, and it's not going to cut it immediately. It's going to cause it to get, first it's going to stop its protein translation, can't make protein. Then it's going to segregate it into a place where it later gets degraded, P bodies. We'll talk about them. But the difference between microRNA and siRNA is this one binds completely, and the microRNA generally has some kind of like these kind of bubbles, these like loops in it, where it doesn't, you got extra bases, so it bubbles that out, okay? So it's incomplete binding. But this is a natural, this was also discovered in C. elegans, but these guys, even though this is like, these things are super important for all kinds of processes, because the C. elegans guys won the Nobel Prize for siRNA, and they're going to use pretty much the same pathway. The people who discovered microRNAs, even though they're super important for development and everything else, are sort of probably messed up. They're probably not going to get the Nobel Prize. But they should, because it's a totally different process, and there's, you wouldn't be able to develop an animal at all unless you had these working properly. All right, so here we're looking at microRNAs. They're just normal genes being expressed as RNA, okay? But they're not protein coding. They're going to form complex patterns based on base pairing, okay? They're going to have a loop at this end like this, and they're going to look sort of like that. They're going to get what's called cropped by a an RNase in the nucleus called drosha. They're going to get exported. Then they're going to get what's called diced, where they get that loop taken off, okay? So now you got two of these, and they're going to get loaded on to a protein called RISC, RNA-induced silencing complex. That RISC protein, so it's a protein complex that's made with argonaute proteins. Okay, so argonaute with this little 21-mer microRNA is going to be the part that's looking for mRNA. When it finds a bind, when it binds it, it's going to take this mRNA and it's going to, its binding is going to block protein, making protein translation. Two, it's going to take it and move it to a P body. Let's move forward. It's going to move it to a P body, which are places in the cell that they accumulate all of these like mRNAs bound to argonaute proteins. And in these P bodies, there's decapping proteins. So you know that the mRNA has a 5' cap that protects it from degradation. That's taken off, and then they get chopped up, okay? So if you look in the cell where the decapping enzymes are, they're in these P bodies. If you look where argonaute proteins are accumulated, because they're the ones that brought it there, they're accumulated there too, okay? Here you can merge, see the same thing, okay? So microRNAs are eventually going to be brought to the P bodies and degraded, okay? But they're not being cut immediately like an RNAi. They're just going to be brought here to these P bodies and then degraded. Okay, so this is another way of drawing it. You've got this microRNAs are initially being cropped by drosha, and then they're diced by dicer, and then they get loaded onto argonaute complexes, which only use one of the strands. Okay, when it gets this incomplete binding, then they're drawing it like here, like they're saying this is the incomplete binding, but that's also not a correct drawing. The incomplete binding is almost always these loops. It's not like part of it, it's a loop. So it's like, but when it forms that loop, like it's a microRNA, it goes to the P bodies where it's going to eventually be degraded. But it doesn't actually cut the mRNA at this point. But if you get 100%, pretty much 100% or so of it binding, then it gets cut. So basically the microRNA pathway is going to have incomplete binding, and it's going to bring it to P bodies to get degraded. And the RNAi pathway is just going to cause it to get cut right there, okay? And this RNAi, as I said, is to block transposon movement, but you can make it against anything you want, and it'll cause the mRNA to get cut. And without that 5' cap, like if you've got a cut here, it'll immediately, what's protecting the mRNA from degradation is the 5' cap and the poly A tail. But when you cut it in the middle, now it's just going to be chopped back because it doesn't have a cap on this side, and chopped back here because it doesn't have the poly A tail protecting it. So it'll be degraded rapidly once you cut it. So this is what it looks like when you have Argonaute proteins bound to the target mRNA. And these are the sites that cut right here for slicing when it's bound. Okay, so there's another pathway by which RNAi can work. So, like I said, you got the RISC complex, that's Argonaute and the siRNA. So siRNA just means short interfering RNA, okay? And it's a 21-mer about, that's the active site. So that's gonna, if it binds completely, that's gonna cause degradation. This occurs in the cytoplasm, generally. But in the nucleus, there's another complex called RITS for RNA-induced initiation of transcriptional gene silencing. This RITS complex is also going to bind to the mRNA, this siRNA. But it's looking for pairs on the mRNA as it's being made by RNA polymerase. Okay? When it gets that link, the RITS is going to cause DNA methylation and histone methylation in this region because it's on this thing, it can bump up with the DNA, and it's going to repress transcription. So almost all your transposons, which this pathway is designed to silence them, they don't want to just keep chopping up the RNA being made all the time. They want to go in, and anywhere there's a transposon that's being made, they want to silence, turn into heterochromatin, that region. So this is actually very important for transposon silencing. But if you're targeting a gene for degradation, it's going to first degrade it, and it's also going to take that region in the genome and repress it. Okay, so it'll shut off two ways. So when this came out in C. elegans, first everybody wanted to work in C. elegans, so we had huge influx of hundreds and hundreds of people because everybody wanted to be able to knock out their gene of interest. And the mammalian people were really stymied. They were like, man, this is such a great thing, but we can't do it. And the reason they couldn't do it is because C. elegans people put in a humongous amount, like 400 or more bases of double-strand RNA. It actually gets chopped up. You get tons of these little target RNAs, these siRNAs, and it works great. It knocks out everything. But if you put in a big piece of double-strand RNA in mammalian cells, the mammalian cells are going to shut down protein translation. Mammalian cells, they've been infected by a double-strand RNA virus. And their first response when they're infected by a double-strand RNA virus, stop all protein synthesis. So in that situation, you can't study anything because, you've basically shut down the cell. But it turns out that these small 21-mers, okay, the small siRNAs, don't induce this block on protein translation. So now mammalian people are like, oh, well, that's fine. We'll just use lots of these little small ones, and we can still target our gene. So that's what they do. So they use siRNA for short interfering RNA, and they either introduce those directly into the cell, or they make a construct, a DNA construct, that has two of these in anti-parallel positions so they can bind each other. And then these things form short hairpin RNA, which also get cut by dicer and cleave to form the same thing. So they get the same about 21-mer siRNA, which then gets put into the RISC complex and can go after target RNA. Okay? So either they're introducing a DNA that encodes the shRNA, short hairpin RNA, or they're directly just adding this to the cell, the siRNA. So because you can, this is pretty powerful. C. elegans is super powerful because it actually, this siRNA effect, RNAi effect, gets spread from tissue to tissue. And so what you can do is you can have bacteria that can't chew up double-strand RNA because they're mutated, and they express this double-strand RNA, so they're basically making it in both directions, and they form large double-stranded RNAs. And so these bacteria are loaded with double-stranded RNAs. Well, these nematodes eat bacteria. They're going to eat it. The intestine is going to take up the double-stranded RNA. The intestine will then chop it up into little pieces and spread it into the rest of the worm. And they actually amplify it also. So they can amplify it also. And then it gets spread out to all the tissues. And you can basically inactivate, like you can do this feeding library where they're eating the double-stranded RNA, and then they shut down the gene in the eggs, right? It gets into the germ line, and then, but they can knock out any tissue they want. So basically there's libraries where if you want to do a screen, you don't have to inject or do anything. You just feed them one particular bacteria, expresses the double-stranded RNA for one gene. You feed them a different bacteria, expresses the double-stranded RNA for another gene. So you can have, so you have libraries, like I have a library in my lab that has like about 16,000 different bacteria that have different genes. And I can just screen by just allowing the worm to eat that, and they'll inactivate the gene. You can say, well, what's a gene good for? And you can inactivate it, right? Like here's a particular gene, you knock it out, and the mitotic spindle doesn't form in the one-cell embryo. Can't form it. So it's a great way of screening for new genes, and then you know exactly what the gene is because that bacteria only expresses the double-stranded RNA for that gene. And you can do similar siRNA screens in mammalian cells, but what you're doing now is you're taking those siRNA, which are these short RNAs, okay, and you've got libraries of them, and you're just introducing them into different wells. So you grow your mammalian cells in the bottom of these wells. Each well gets a different siRNA, and you can because there's so many wells, by the time you screen through the whole genome, you need to have the thing automated. So it's got an automated imager that just like moves it and takes pictures of every well. And then let's say you were interested in, like mitotic spindle. So now you can knock out, you can say, oh, the mitotic spindle doesn't form two, it forms monopolar or multipolar, or it doesn't have asters or whatever. And you can just do screens for whatever you're interested in if it occurs in tissue culture. So in this way, you can also do genetic screens by knocking out all these genes in mammalian cells also. All right, so we're moving on. We only have a few minutes left, so we're going to go with all right, so we're gonna start with CRISPR-Cas9 because it's in the news a lot. So this is the most powerful genome editing approach to date, and people just like the politicians don't even understand what's happening because there's no limitations on it yet. But you can do almost anything you want with it, as we'll see. So it's naturally a bacterial defense mechanism. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. So what these are regions in bacteria that they see these sequences one after the other, and they have a set spacer in between, so they can tell these are repeats. Okay, but each one of the repeats is different. And what's going to happen is the bacteria, when they get infected with like a phage, a virus, or plasmid DNA, they're going to cut it, and then they're going to turn it into one of these CRISPR repeats, okay? So, like if you get a plasmid DNA introduced, it gets cut, and then turns into one of these CRISPR repeats with sequence in between that tells it how to use it, okay? But it's basically, which allows it to load into these DNases called Cas. So these Cas genes are nucleases that they're gonna cut DNA. They're gonna bind to a guide RNA, and they're gonna target whatever that guide RNA tells it to cut. Okay. So again, so the reason these things are arising is, let's say that you got some plasmid coming in. The bacteria is like, what is this plasmid? I don't want this plasmid. It's going to cut it, and it's going to put it into this CRISPR repeat. The Cas9s are always being made. There's lots of them. People like number 9, and so number 9 works well, and that's why it's called CRISPR-Cas9. But now, because people are using different Cases or different types of enzymes, they're generally just calling the whole system CRISPR. So what happens is then each one of these things is then being expressed along with extra sequences on the end. That extra sequence allows it to get embedded into the Cas proteins, like Cas9, and then it'll chop up at whatever sequence they took out of the plasmid, it'll cut it at that sequence. And it'll work for a virus too, a double-stranded RNA virus. So this is a defense mechanism where if a strange piece of DNA comes in, it's going to take it, put it into the CRISPR array. That CRISPR array will get turned into RNA. The RNA will get loaded into the nuclease, and it'll use that as a guide RNA to target what it wants to do. And you could say, well, why doesn't it target these sequences, right? Because there's a whole mess of them, right? And it's looking for those sequences. But it can't target these ones because it uses this thing called PAM. So PAM is one of the things that it's looking for. And the PAM is like this NGG. Then this thing binds here. So your guide RNA is going to bind to the sequence, but it wouldn't even see that sequence except for this sequence here, the PAM. It's usually NGG, it can be GGG, it can be other things. The Cas9 protein looks for the PAM sequence. When it sees the PAM sequence, then it says, well, do I have a match? So if it doesn't have the PAM sequence, it won't do it. Well, the PAM sequence, while it's in the target that's going to get cut, and it was in the original target, okay, is not in the repeat part that's put in the genome. So while the PAM was there, because that's why I chopped that section, right? It's now only in the target. It's not in the repeat. Okay, so it's a sort of smart way. It needs to have this sequence. It was there already, but it doesn't put it into its guide RNA because this guide RNA came from that repeat, right? Like you got, this spacer and then another repeat and a spacer, another repeat like this, right? So these are the repeats, but there's no PAMs here. Okay, so it won't chop up this CRISPR repeat. So everybody see that? Okay, so all right, so how is this being manipulated? It's being manipulated like this. The only thing they're doing is it allows you to cut almost anywhere you want in the genome. You're like, well, how powerful is that? Well, it's powerful because, one, you can knock out genes by cutting them. You're like, oh, the cutting knocks it out. Well, not really, because it's going to get repaired. But the repair is going to be through non-homologous end joining. In the non-homologous end joining, you always chew back some of the sequence before you join them together. Okay, so they're going to lose some sequence, and if you lose an odd number of bases, so it's not odd, but not a 3-mer, then suddenly it's out of frame. Okay, you're reading it every 3, but now it's out of frame, and then that's the end of that protein, right? It'll go out of frame from that point. So one, you can knock out any gene you want, but then you can also use homologous recombination to come in and introduce whatever you want. So let me just draw it really fast before we leave. Let's say I cut here. I'm going to come in with homology arms on this side, and then I can introduce anything I want in this place. It could be the exact sequence where I've just mutated a few bases. I could put in a whole nother gene. I could cut it. I could cut this one here. I could cut it here, and I could come in with homology arms over here and over here to delete this whole thing, or I could put in another thing. So I can start moving things around by wherever I put the homology arm, it's gonna recombine in, and I'll put in whatever I want in between. So it's very powerful in terms of manipulating the genome. And all it's doing is just a way that you as whoever you are, like generally scientists right now, hopefully it doesn't get to the level of just people doing it in the garage, can cut whatever they want and insert whatever you want. Like maybe in the future you're like, oh, I want a blue eye color. I'm going to knock out in my children, I'm going to knock out the brown pigment gene. That's easy to do. It's easy to knock out genes, but you can also put things in. You can also say, oh, this person has a genetic disease because they have these mutations in that critical gene. I'm going to cut it. I'm going to put in the right sequence or whatever you want. I want my child to be stronger. I'll affect growth hormone levels. I'll do what I want until it gets regulated. But it may not be regulated, But it's coming. So anyway, that's it. So the rest of this thing won't be on the exam, only up to CRISPR, and the rest of it's not on there. And review session 2 days.